Mesdames et messieurs, veuillez accueillir vos coprésidents du congrès de l'ACRV 2023, les docteurs Jerome Estaquier et Cecilia Costiniuk. Ladies and gentlemen, please welcome to the stage your CAHR 2023 conference co-chairs, Dr. Jerome Estaquier and Dr. Cecilia Costiniuk.
Oh purée, ça j'aime pas. Alors, c'est moi qui parle. Bonjour à tous. Vous voyez, en fait, on va être un binôme, un français et une anglophone. Alors, chers amis, en fait, on est aujourd'hui, et au nom du comité d'organisation, Cecilia et moi, nous sommes en fait extrêmement heureux de vous revoir tous aussi nombreux. Aujourd'hui, 675 personnes sont présentes, 350 posters ou résumés ont été posés à la 32e réunion canadienne sur la recherche sur le VIH. Cette conférence est un peu particulière parce qu'effectivement, la dernière fois qu'elle s'est organisée à Québec, c'était il y a 17 ans. J'étais pas là. Vous l'avez tous reconnu à mon accent, effectivement, je suis très québécois. Donc nous sommes contents, en fait, pourquoi aussi de se retrouver, puisqu'effectivement, au cours de ces quelques années, le confinement, en fait, nous a un petit peu cloisonnés et réduit presque au silence. Et j'ai un mot en particulier pour les étudiants, puisque souvent, finalement, ces réunions plénières sont pour eux l'occasion, en fait, de se réunir et de discuter. Et je vais laisser ma parole maintenant à Cecilia, qui va vous le faire en anglais, plus court.
So, welcome. So, welcome everyone to Quebec City for the 32nd Canadian Conference on HIV/AIDS Research. After a very long pandemic, it gives us great pleasure to be reunited with you all today in person, and we're very pleased to announce there's over 675 participants and over 300 abstracts.
Alors maintenant, en fait, on est également ravis puisqu'on va pouvoir accueillir Éric Picard, représentante en fait de la communauté de Wendake. Merci.
We are now honored to welcome Madame Edith Picard, an elder from Wendake amongst us, who will lead the opening of the ceremony.
Bonjour. Bienvenue sur le territoire du Neo-NCO. Rappelons-nous que nous sommes responsables des différents territoires où nous vivons et que nous fréquentons. Nous sommes de différents horizons. Nous sommes de l'Est, du Sud, de l'Ouest ou du Nord. Nous sommes réunis pour un moment d'échange, de collaboration. Afin d'être en harmonie, afin de faire union, je nous invite à rendre grâce. Rendons grâce au Créateur. Remercions pour la terre-mère, pour l'eau, le feu, l'air, pour le soleil, la lune, les étoiles. Remercions pour tout ce qui vit sur terre, ce qui nous nourrit et nous maintient en vie. Remercions pour nos ancêtres et pour tous ceux qui nous aident dans notre vie. Rendons grâce pour les enseignements de la nature, pour les dons et talents de chacune, de chacun. Je tiens à exprimer ma gratitude pour le privilège d'être parmi vous depuis ce matin. De la gratitude également pour toutes ces personnes qui, de près ou de loin, ont travaillé à la préparation de la conférence. Et toutes ces personnes qui, soit dans votre milieu de travail, votre milieu familial, ont contribué à votre présence à la conférence. Avoir reçu une invitation ou avoir choisi de s'inscrire implique un intérêt, une préoccupation, une sensibilité et une volonté de contribuer à la santé, à un avenir. C'est une mission qui nécessite une mise en commun des aptitudes, des forces, des spécificités de chacune, de chacun. Osons personnellement nous faire confiance. La nature est faite de diversité et de contraste, c'est ce qui fait sa beauté, sa grandeur. Rendons grâce d'avoir une occasion de partager nos réflexions. Demandons la lucidité, la clairvoyance pour faire des choix prioritaires et identifier des solutions réalistes, voire même innovatrices. Je vous souhaite une très belle conférence, de la collaboration, de l'implication, de l'écoute, une participation indispensable. Tiaweng, merci pour votre présence. Merci pour votre engagement. Bonne conférence. Je vais me risquer en anglais. Kwe, hello. Welcome to the territory of Neo-NCO. Let's remember that we are responsible for the different territories we live in and visit. We are from different backgrounds, different contexts. We are from the east, south, west, or north. We are gathered for a moment of exchange, of collaboration. In order to be in harmony, in order to be united, I invite us to give thanks. Let us give thanks to the Creator. Let us give thanks to Mother Earth for the water, the fire, the air, the sun, the moon, the stars, for all that lives on earth, for what nourishes us and keeps us alive. Let us give thanks for our ancestors, for all those who help us in our lives. Let us give thanks for the teachings of nature, for the talents of each and every one. I would like to express my gratitude for the privilege of being among you since the morning. I am also grateful for those people who have worked in the preparation of the conference and all those people in your work or family environment who have contributed to your presence at the conference. Having received an invitation to participate or having chosen to register implies an interest, a concern, a sensitivity, and a desire to contribute to a healthy future. A mission that requires a pooling of skill, strengths, and specificities of each and every one. Let us personally dare to trust ourselves. Nature is made of diversity, of contrast, which makes it greatness, its beauty. Let us give thanks for the opportunity to share our reflection. Let us ask for lucidity, foresight to make priority choices, and identify realistic, even innovative solutions. My best wishes to each of you for a good conference, collaboration, involvement, listening, and indispensable participation. Thank you. Tiaweng.
vous avez tous vu le titre, tel un boomerang. En fait, ce titre a été choisi par le comité et qui a réfléchi en fait, à une vision, qui avait une vision commune en fait, de ce qu'on voulait un petit peu au niveau du colloque, ou en tout cas de son meeting. Car effectivement, en fait, tel un boomerang, en fait, on est allé du sida vers la Covid et aujourd'hui de la Covid au sida. Et je dirais qu'il n'y a qu'un pas, parce qu'effectivement, lorsqu'on regarde la pandémie par rapport à celle du VIH, eh bien, en fait, la Covid a été finalement un catalyseur à la fois aussi bien pour la recherche scientifique, la recherche clinique, l'épidémiologie et sociétale. Et ce, finalement, grâce à nos leçons que nous avions tirées concernant, depuis plus de 40 ans maintenant, nos connaissances sur le VIH, et qui malheureusement restent toujours une question d'actualité. Et ainsi, des objectifs sous-jacents finalement à cette journée, c'est de faire un peu l'état du progrès de ces connaissances qui, aujourd'hui, par la Covid, en fait, nous amènent à une nouvelle vision, peut-être, de nouvelles avancées. Et il faut mettre à profit, finalement, ces connaissances que ce Covid nous a apportées pour mieux comprendre, peut-être, les questions qui demeurent et qui demeurent toujours aussi essentielles. Pathologie, absence de vaccin. Et pour autant, ces questions demeurent. Par conséquent, en fait, nous devons tous nous mobiliser pour atteindre le fameux 90-90-90 à nouveau. Et enfin, tout ceci nécessite quoi? Aussi des investissements. On a vu combien l'investissement était important sur la Covid. Nous avons besoin de l'ensemble des acteurs, aussi bien sociétaux que politiques, mais aussi des chercheurs. Je vais laisser à la main à ma confrère.
So appropriately, our conference is entitled A Boomerang Effect from HIV to SARS-CoV-2 and Back Again. Like the HIV epidemic, the COVID pandemic has been a catalyst for scientific advancement, and lessons learned from HIV have contributed to this progress. An underlying goal of this conference is to reflect on both advancements and setbacks during the pandemic and lessons learned. Furthermore, we will refocus ourselves to re-tackle the 90-90-90 goals with new perspectives. Fighting pandemics requires investment, innovation, and a strong commitment from everyone. So along this vein, we would like to thank the CAHR Scientific Committee members who have worked tirelessly to bring you this outstanding program.
Et donc, à ce titre, en fait, on voudrait remercier d'une part, effectivement, les organisateurs du CAHR, son président, Kev, bien entendu, les autres membres, Andrew, Viviane, Erin et Andrea, nos collègues et amis qui ont participé à l'organisation de ces 3 ou 4 journées très intenses, la recherche fondamentale avec Petro et Ali, Marie-Louise et Sébastien pour la partie clinique, l'épidémiologie avec Marie-Pierre et Souleymane, Gilbert et Patrick Labbé pour la partie communauté et santé publique. Merci à vous tous.
We would like to thank the CAHR committee, including Andrew, Vivian, Aaron, Ian, and Andrea for guiding us through the process of putting together this scientific program for you. We'd also like to thank our friends and colleagues, as well as the individuals listed here on the program planning committee. So from basic sciences, Dr. Ann Kuta and Dr. Janabian. Clinical Sciences, Dr. Marie-Louise Vachon and Dr. Sébastien Poulain. Epidemiology and Public Health, Dr. Marie-Louise Gagnon and Dr. Souleymane Diabat. Social Sciences, Dr. Gilbert Raymond, and our community representative, Mr. Patrick Labbé. We would also like to thank the friends and colleagues and innumerable volunteers who have contributed to the success of this program.
Donc nous allons accueillir maintenant, si je ne me trompe pas, financial support. Ah oui, très important. Sorry. Non, on ne rigole pas. C'est sans le support du CAHR, bon nombre d'entre vous ne pourraient pas être là. Je pense en particulier aux étudiants, aux personnes de la communauté, aux fellowships. Et c'est encore une fois un point important puisqu'on était dans une zone de confinement. Donc le CAHR, de ce côté-là, merci encore.
So we would also like to recognize CAHR for the financial support provided to the next generation of researchers and members of the community who are attending the conference thanks to scholarships that facilitate participation in this important event. There have been almost 80 recipients of community registration scholarships and 8 community travel awards.
C'est vrai qu'on doit les aider. Donc maintenant, on accueille Kate, c'est ça? So, maintenant, on va accueillir... Non? Non? Ah oui, mais moi, je vois rien du tout. Alors, ah oui, c'est ça. Donc, nous allons... Parce qu'en fait, moi, je suis trop petit. Hein. I'm too small. So, first, we have thanks Basic Science.
So, Basic Sciences, Charmaine Bégun and Alexis Chacon.
Clinical Science.
Wassal El-Greti and Joanne Brochon.
Epidemiology and Public Health Science.
Yasaman Sadeki and Rodrigo Alejandro Sierra Rosales.
Social Science.
Tricia Campbell and Jason Lohog Tian.
And now, Keith. So I'm not. Je ne vais pas parler en français, je vais laisser simplement Cecilia parce qu'il ne comprend pas le français. Alors, on va pas switcher en anglais, ça sera plus simple.
Okay. So now it's a pleasure to introduce Dr. Keith Fouke, President of CAHR. Dr. Fouke is professor and head in the Department of Medical Microbiology and Infectious Diseases at University of Manitoba. His lab focuses on defining cellular immune mechanisms of natural resistance to HIV and the control of infection if it does occur. His current studies include preventing HIV infection by reducing inflammation of the genital tract. Dr. Fouke has a particular interest in training young investigators in multidisciplinary research, and he has been involved in a number of workshops and international training programs.
I come bearing gifts. Bonjour tous. Thank you, Jerome and Cecilia, and thanks to for your leadership in this conference and to the whole organizing committee. Look forward to a great conference. As president of CAHR, it's my honor to take part in the official opening of the 32nd CAHR conference, and to do it in 3 dimensions, not on Zoom, not on Teams. Those formats have some use, but you can't hug a computer. Well, you can hug a computer, but it doesn't hug back. That's the problem. So it's great to be here in person. There's been a lot of ancillary events and satellite sessions, and I've been here all week, and it's been great to connect with a lot of colleagues that I haven't seen for 4 years. And so it's wonderful to have that personal connection again. And that's why we come to these meetings. There's still 3 more days to go, and I look forward to the great scientific session. To begin, I'd like to take the opportunity to recognize the CAHR members of the board of directors, and I'd like to thank them for their hard work and volunteer commitment for helping CAHR help navigate CAHR through the pandemic over the last several years. It hasn't been easy, but they've done a fantastic job. So I'd like to thank all of our CAHR board members, and let's give them a round of applause. And of course, this meeting wouldn't be possible at all without our sponsors, so I'd like to take a time to appreciate the sponsors, many of whom have stuck with us through the pandemic, and without their generous contributions each year in and out, CAHR would not have been able to host this great conference or to even have been able to survive over the last few years. So it's great that we have such great support from our sponsors, and I encourage all of you to visit them in the exhibit hall. And so the sponsors are listed there. The annual CAHR conference has been the premier gathering event in Canada for those working in all disciplines of HIV research, as well as policymakers, persons living with HIV, and so many who are committed to ending HIV. It's great to be here in Quebec City for CAHR 2023. We were supposed to be here in 2020, and then, I don't know, something happened, and we went virtual, and it's great to be back here in person. It's such a beautiful city, and it's the home to so many very talented researchers, many of whom you'll be able to see their work over the next few days. Overall, CAHR 2023 will be a tremendous opportunity for all researchers and community members across the country to share the latest scientific advances in the field and learn from one another's expertise and develop new ways to treat and prevent HIV. So I hope all of you will enjoy the conference. Thank you for making this a priority in your busy schedules and for your contributions. So thank you. And now I'd like to invite Mr. Andy Pringle, who's the chair of the CANFAR Board of Directors, to join me on stage. Andy will help me present the winners for this year's CAHR CANFAR Excellence in Research Awards. But first, Andy is going to provide some comments about a colleague, Chris Bunting.
Thank you, Keith. I'm indeed honored and privileged to be here to say a few words about my friend and colleague, Chris Bunting, who was one of the leaders, builders, and contributors to CANFAR over such a long period of time, and who tragically passed away earlier this year from cancer. Chris was a relentless advocate for AIDS research, for LGBTQ2S+ rights in Canada. He was an esteemed colleague and board member of our organization, and he was involved with CANFAR for over 30 years, doing a wide variety of roles, but he, from 2010 to 2017, was president and CEO, and from 2017 till his death was the deputy chair. Now, Chris and I met when we both came on the board in 1992. We became good friends, and about 3 years after that, I introduced him to my wife's brother, and they were partners, Chris and Anthony, or Tony Whittingham, as he's publicly known, were partners for throughout that time for the last 25, 30 years. So Chris was not just a colleague and a friend, but he was part of the family. Chris had an illustrious business career. He was an alumni of both Carleton University and the University of Ottawa, and he went on to be president and chairman and CEO of the renowned PR firm Weber Shandwick, which advised top 500 companies in business strategy and enterprise communications. And Chris also acted as a leader and a mentor to hundreds of employees, clients, and friends over many years. Chris moved to London, England in 2002 to become the senior managing director for a global consulting firm, Inzio. And he also became, later in his time in London, became the chief executive officer of the Awareness Foundation. In 2010, Chris and Tony returned to Canada, and Chris engaged in a number of enterprises and not-for-profits that he felt passionate about. But as I said, he took on the role of president and CEO of CANFAR, helping drive the CANFAR's mission to end HIV in Canada, providing strategic input and expert guidance to the organization, its board of directors, executive team, as well as employees and volunteers. Chris was a mentor and coach to many business leaders, an esteemed advisor to colleagues in the LGBTQ2S+ community and HIV organizations and advocates across the country. He's also a passionate believer in the value and importance of research in the fight against HIV and AIDS in Canada. And he was also a very strong believer in the work of this organization and its members. And I know he would be justly proud of the recipients today and the work they have done and will do in the future. Thank you.
Great, and Andy will help me present the awards. And thank you, Andy, for your service to the community through your leadership at CAHR. I'll just tell you a little bit about the awards. The CAHR Excellence in Research Awards highlights and celebrates the contribution of Canadian investigators in HIV research in Canada and internationally. And I'll just point out that these careers are targeted, these awards are targeted at mid-career investigators, because that's often a time when there's sort of a lull in recognition for researchers. And so thanks to the CAHR board and also to CANFAR, these awards were developed. And I can tell you they're highly competitive awards. The recipients of the awards have a proven track record in excellence and have demonstrated a commitment to improving the lives of those living with or at risk of contracting HIV. And before we pay tribute to the 2023 winners, I'd like to offer a belated but wholehearted and in-person congratulations to the winners from 2021 and 22. See, if you get to win these awards, then the next year you get to be chair of the conference. Right, Cecilia? So congratulations to those individuals. And now, as I read the names for the CAHR 2020, the 2023 CAHR CANFAR Excellence Awards, we'd like these awardees to come up and we'll receive their prize and we'll take a photograph. So I'll give their names. We'll acknowledge each of them through a round of applause, and then they'll come up, but they can come up one after another. You don't have to wait until the other person is done receiving their award. So, for the 2023 award winners, in the basic science category, Dr. Sabrina Brumet from Simon Fraser University. Yeah. Yeah. You're wearing green. Safer picture. Under the clinical sciences category, Dr. Bertrand Luboucher from McGill. In the epidemiology and public health category, Dr. Nathan Lakowski from University of Victoria. And in the social sciences category, Dr. Carmen Logie from the University of Toronto. And there's a fifth category for the community-based researcher, but unfortunately the researcher is not here. But I'd like to acknowledge that the winner is Dr. Mary-Lou Gagnon from the University of Victoria. Okay, great, and the awards just keep on rolling. Now I have the pleasure of announcing the CAHR Red Ribbon awardee for 2023. Established in 2001, the Red Ribbon Award is presented annually by CAHR for outstanding service to the cause of research in a way that has increased our understanding of the tremendous, understanding of treatment and prevention of HIV/AIDS, while enhancing the quality of those living with this disease. Before we pay tribute to the 2023 winner, I'd just like to offer a belated but heartfelt congratulations in person to those from 2021 and 2022 winners, Ron Roseness and Maureen Owino. On behalf of the CAHR Board of Directors, it's my extreme pleasure to announce that the 2023 CAHR Ribbon awardee is Joanne Lindsay. Unfortunately, Joanne isn't here in person to be able to accept it, this award. So accepting the award on her behalf are Joanne's nominators, Mona Lifty, Anne Burchill, and Abigail Croach. Please come up.
So Joanne was unfortunately unable to be here today to accept the award, but she would have loved to have been here. She's unwell. But we thought we would take a few minutes to talk a little bit about why we nominated her. And then there is going to be a film played by, that was recorded by Joanne's adult children, Charlie and Chiku Lindsay, who are going to say some words on Joanne's behalf. So I want to just take a moment to say why we nominated Joanne. Joanne's been an enthusiastic community research champion for well over a decade. She's a strong advocate for research among women with HIV, mental health, and cancer prevention. She's had many roles in research, including as a peer research associate, a co-investigator, a lead of knowledge translation activities, chair of community advisory boards. And one of the ways I've come to personally know Joanne most is through her dedicated efforts to improve literacy about human papillomavirus or HPV-associated cancers among women living with HIV. On her own initiative, Joanne developed a workshop for women to learn about HPV-related cancer, to encourage them to get screened, and to motivate them to get involved in the research. With amazing energy, she's organized and led many workshops and talks on this topic, including to the UNAIDS Civil Society Advisory Group in 2020 and at AIDS 2022. Joanne's also a mentor to early career researchers to help develop their skills in community engagement, and they talk about her as having been instrumental in their growth. She's volunteered countless hours to serve on committees and boards to advance research in KT for people living with HIV, including being on the KT Board of Directors, on CHASRAC, the CAHR, HIV and AIDS and STBBI Research Advisory Committee, and the Canada International HIV and Rehabilitation Research Collaborative. Joanne does all of this with a special grace and a smile. She's genuinely engaged, she asks important questions, and brings valuable insights from her lived experience on women's issues. When we prepared our nomination, we asked people to describe Joanne, and they commonly used words like advocate, curious, generous, impactful, a leader, tireless or tenacious, and a translator of research findings and a teacher.
Joanne served as a board member on the Ontario HIV Treatment Network and on the governance committee of the OHTN cohort study. As part of the cohort study governance committee, she was also a co-chair and provided insight for researchers and for community members to understand protocols. She was a very committed, active, generous member of the board. She served on both finance and governance committees and the scientific review. She brought her experience as a woman living with HIV and as a peer research associate. She was actively involved in helping the OHTN identify research priorities and support peer researchers and young emerging researchers. Highly connected with other women living with HIV, she was able to bring their issues to us and the findings of research relevant to them. She is also just an incredibly lovely person who always speaks her mind.
Thank you, Anne, for leading this well-deserved nomination for Joanne. I'm sure many other people in the room would have joined us in nominating her. I personally know and work with Joanne as she is one of the first peer community research associates and co-creators in Canada, one of the first cis women community peer research associates in Canada. She really has been a role model to researchers, to trainees, as well as to many cis and trans women and non-binary folks and all the people living with HIV to be involved in research. I had a chance over the past few days to speak to several women living with HIV who have been inspired by Joanne to get involved in research. And this is what they said. Thank you, Maureen, and others. Joanne has always advocated for women living with HIV to be leaders in HIV. Joanne has been and is a huge leader in the capacity building of women living with HIV, and this leadership cannot be ignored. We, as women living with HIV, acknowledge her wisdom, expertise, and her courage. Thank you, Joanne. And just to add, that she always did all of that, or has done all of that, with a warm smile and compassion and kindness. And it's hard not to cry. And so now we're going to watch a video from Joanne's Joanne's children. And we're like,
hi, I'm Chiku. This is my brother Charlie. We are Joanne Lindsay's children.
Yep, our mom is not able to do her speech in person, but she did write one and she wanted us to deliver it. So here it goes. Hopefully we do it justice. I'm deeply honored to be nominated for this award, and I accept this award with great humility. I thank those who nominated me, Dr. Anne, Dr. Kelly, Dr. Mona, and Dr. Abigail. I ask that they accept this award on my behalf, and I'm sorry I'm not able to be there in person as I've been in and out of the hospital for the past month. I want to dedicate this award to the women I've worked with over the years in peer and support groups. I have learned so much from them, and I hope they have learned just as much from me. Now I'll pass it off to Chiku.
We are so proud of our mother for all the hard work and dedication she's put into HIV research. I have seen firsthand the time, effort, and excitement she has learning and collaborating on research initiatives. Our mother has been involved in HIV research for over 12 years, and educating others has been a particular strength and passion of hers that has hopefully inspired others to learn more about HIV. Joanne has an excitement for learning, and through her work at PWA, Katie, Positive Women's Network USA, Casey House, and the Estimate Lab at St. Michael's Hospital here in Toronto, she's channeled that excitement into valuable education. Congratulations to our mom, Joanne Lindsay. We are so proud of you and everything you've accomplished and are incredibly lucky to have you as a mother.
Yep. Congrats, Mom. Yeah, I think just to add on to what Chiku said, Thank you. Our mom is super passionate about HIV research, and she's really jumped headfirst into this community for the past however many years, several years, and yeah, she truly has a passion for it, a passion for learning, and to just be able to go to some of the places she works and to see the community that she's a part of and the people she works with and the joy that it brings her, it truly is inspiring for both of us and our whole family. So from the bottom of our hearts, thank you very much. We love you, Mama. Get well soon. And yeah, so proud, girl. You did it. You did it.
We love you, Mama.
Mwah. Thank you, guys. Appreciate it.
Mark Wainberg. Donc il est temps pour nous, en fait, avec Cecilia, d'ouvrir donc cette session Mark Wainberg 2023. Mark a été un pionnier en recherche, comme vous le savez tous, un vivant ardeur des défenseurs et activiste des premières heures, développé la santé publique pour tous. Et il y a désormais pratiquement 40 ans, cette maladie émergente, à l'époque, on m'a dit de travailler sur le sida parce que j'avais fait un cours de champignons, à tel point que 40 ans plus tard, je suis toujours à faire du sida, et pour laquelle, finalement, nous n'étions pas préparés. On dit toujours, on pense qu'on l'est toujours, mais Covid nous a montré qu'on ne l'était jamais. Maladie stigmatisante et fatale, eh bien, Mark a participé, finalement, à contribuer à révolutionner notre approche médicale, clinique, virologique, concernant toutes ces approches également politiques, pousser le politique là où il ne voulait pas aller. Cette nouvelle épidémie, donc, nous a permis aussi de montrer aussi quels étaient effectivement l'apport et la contribution de Mark, finalement, à ce qu'on prenne en considération aujourd'hui la Covid. Ces approches de Mark a été effectivement le développement de nouveaux traitements, vous savez tous, pour le 3TC, mais il a été aussi un formidable formateur. Plus d'une centaine de personnes, pousser les gens à les éduquer, les former, et on en aura un dans quelques minutes. Eh bien, effectivement, Mark a été un petit peu ce leadership, initialement donc directeur de recherche de l'université de McGill, à l'hôpital juif, et puis il a été finalement donc ce fameux leader. Eh bien, finalement, bien que Mark nous ait quittés en 2017, déjà qui est presque 5-6 ans, eh bien, Mark, en fait, vit toujours grâce, finalement, à quoi ? Grâce au CAHR. Et il faut quand même remercier le CAHR pour cet acte-là et cette reconnaissance de Mark.
So since 2001, the CAHR Conference Scientific Committee has bestowed the honor of the Mark Wainberg Lecture to a Canadian researcher to both honor Dr. Wainberg's contribution and to recognize the efforts of others in the research community who exemplify the same traits of excellence, perseverance, and commitment to the cause of finding innovative and groundbreaking ways to address the epidemic.
Donc aujourd'hui, nous avons la joie et finalement l'honneur d'accueillir en fait et d'introduire Éric Cohen. Et je dirais pour ce cours magistral, puisqu'effectivement, en français, ça se dit un cours magistral et une lecture, c'est un cours magistral, sur les interactions entre le VIH et son hôte, mais aussi de tout ce qu'il nous a permis d'apprendre, mais tout ce qui reste encore à apprendre. Éric donc a été finalement ce jeune étudiant qu'il a rencontré lorsqu'il était en master, étudiant à la maîtrise, pour être en français. Et dans les années 80, donc, il a rencontré Mark dans le cadre d'un cours de virologie. Et finalement, pratiquement depuis 30 ans, Éric travaille en virologie sur le VIH. Comme quoi, finalement, avoir des mentors, ça sert à quelque chose, des chefs de file en québécois, ou des patrons, comme on dirait en France, mais qui ont une connotation un peu très hiérarchique. Éric est donc directeur du laboratoire de rétrovirologie humaine à l'IRCM, professeur de virologie à Montréal. Il est aussi responsable du programme CAHR, où pour un certain nombre d'entre nous, ils sont inclus, dans lequel on étudie la question des fameux réservoirs viraux, développer de nouvelles stratégies pour améliorer, éradiquer, ou en tout cas de chercher à éradiquer ce virus. Et donc Éric travaille sur ces questions-là. Il est membre de la Société royale du Canada et de l'Académie canadienne de sciences et de la santé. Et donc c'est avec un grand plaisir que j'accueille Éric, mais d'abord, place à l'anglais.
So today we have the pleasure to introduce you to the 2023 Mark Wainberg Lecturer, Dr. Éric Cohen, whose life was first touched by Mark when he was a young graduate student taking his very first virology course in the 1980s, and who inspired Éric to pursue work in the HIV field. Dr. Éric Cohen is now head of the Laboratory of Human Retrovirology at the Montreal Clinical Research Institute, ERCM, and professor of virology at the University of Montreal. Since 2014, he leads the Canadian HIV Cure Enterprise, or CAHR, which is a multidisciplinary research consortium dedicated to the study of HIV reservoirs and the development of effective therapeutic strategies towards HIV remission. Dr. Cohen has been involved in HIV/AIDS research for the past 3 decades, and his current work focuses on defining HIV-host interactions regulating viral replication, transmission, and persistence, and on developing new therapeutic strategies to target persistent HIV reservoirs. So we now welcome Dr. Éric Cohen.
So I would like to first thank the organizers, and particularly Cecilia and Jerome for their kind invitation to give the Mark Wainberg Lecture this year at the CAHR conference. I'm going to, so I don't have any conflict. So I'm going to go very quickly through the learning objective later on when I'm going to go through the outline of my presentation. So c'est vraiment tout un honneur de donner cette conférence car Mark Wainberg a été pour moi un mentor, comme l'ont introduit Jérôme et Cecilia. Il a été également un mentor et un collaborateur et un ami. Mark was not only a prominent HIV scientist, but he was also an exceptional human being. He was known for his kindness, compassion, generosity towards his colleagues, and importantly, his advocacy for people living with HIV. And these qualities endear him to many of us that had the privilege to work with him. So it is for me a privilege and an honor that, to give this lecture that was established to underline Mark Wainberg's many contributions in the fight against HIV/AIDS. So what I would like to do today is really present an overview of the advances that have been made in investigating HIV-host interactions that govern HIV replication and persistence, since this has been the focus of my scientific program in the last 30 years. I will first go through the early days with the discovery of HIV accessory protein and the key insight of the HIV life cycle. I will then discuss how the mapping of HIV-host interaction has really changed the paradigm about HIV replication and persistence. I will then talk about these host antiviral factors that are present in HIV target cells and how they are counteracted by specific protein of the virus called that accessory protein. And along the way, I will highlight the implication of these advances in drug development and HIV cure. So I started to work on HIV in the mid-1990s. The epidemic was rapidly spreading across the world, with approximately 10 million people infected. Only one HIV anti-drug, AZT, was available, providing a bandage in an hemorrhage. The survival rate of a person after an AIDS prognosis was less than 2 years. This was a very dark experience as a young scientist, especially since a large proportion of people dying of AIDS had my age. So following the isolation of human retroviruses that we know now is HIV in the early 1993, the molecular cloning and subsequent nucleotide sequencing of the genome in 1985 really led to the realization that HIV was different from the retroviruses that we knew then. In addition to the GAG, POL, and ENV genes that are encoded by all retroviruses and that specify viral structural protein as well as enzymatic function, HIV was encoding regulatory genes like TAT and REV, as well as a series of open reading frames that were initially designated by alphabetical letters, notably the R and the U open reading frame. So my main project as a postdoc was to analyze the HIV genome and to assess whether these open reading frames were encoding a novel class of viral proteins. These studies led to the discovery of protein encoded by the viral protein U and viral protein R genes. And here, I cannot pass without sharing with you the little story about this discovery and the unexpected help of a well-known fashion designer. When I arrived in the lab of Bill Hazeltine at the Dana-Farber Cancer Institute in Boston in the mid-1986, we had the visit of Ralph Lauren. The fashion industry was heavily affected by the AIDS epidemics, and Ralph Lauren wanted to help by directing the usual Christmas and New Year present he was given his business partners, employees, and customers to AIDS research. With these unexpected funds, which were significant at the time, we initiated a program that no public organization would have funded. We designed small peptides corresponding to the amino acid sequence derived from all open reading frames that could be identified throughout the HIV genomes and generated antibodies against the putative proteins. It may be obvious nowadays, but in the early 1990s, it was a huge undertaking. So following in vitro transcription and translation, and at the time there was no kits, we were able to demonstrate that these ORF, these open reading frame, were expressing viral protein recognized, as it's shown in the notes of my lab notebooks at the time, by serum from HIV-infected individuals, and that they could be detected in T cells upon HIV infection. And it is with these tools that we were able to discover VPU, then VPR, the 6 and 7 gene encoded by HIV in addition to VIF and NEF. So as a group, these viral genes and the protein that they are encoding were named accessory proteins because very early on, we showed that they were dispensable for viral replication, at least in vitro. So while the VPR gene was encoded by all primate lentiviruses that were subsequently isolated and sequenced, and VPR is shown in red, VPU was only encoded by HIV-1 and the simian immunodeficiency virus from chimpanzee. It was not encoded by HIV-2 and SIV from macaque, which encoded a protein called VPX that has nothing to do with VPU. And this pointed to the fact that SIV chimpanzee was very likely the precursor of HIV-1. It also suggested that the very distinct pathogenesis and global distribution of HIV-1 relative to HIV-2 that was really endemic resulted, at least in part, from differences in the accessory protein that they were encoding. So in order to put the function of this accessory protein in context, let me first go through an overview of the HIV-1 life cycle that was the subject of a lot of work during the first decade after the sequencing of HIV. As you know, HIV infects primarily CD4 T lymphocytes and macrophages, and it enters into these cells through interaction of its envelope glycoproteins with, it enters into the CD4 T cell and macrophages via sequential interaction between the viral glycoproteins with CD4, the primary receptor, and either CCR5 or CXCR4, the two main co-receptors. Following entry, the genome undergoes reverse transcription to a double-stranded form. The DNA genome is then transported to the nucleus, where it is integrated into the host DNA, and there it becomes essentially like a host gene. And this is the ultimate way for the virus to persist. Following that, viral genes are expressed by host transcription factors and the cellular translation machinery. New viral particles are assembled and released from the cell surface, and the virus undergo a last stage, which is a proteolytic maturation of the viral particle, which gives it its infectious potential. So these basic studies led to a detailed insight into the HIV life cycle, which allowed the identification of the targets of the antiretroviral drugs that have been used in combinations since the mid-1990s and that had such a transforming impact on HIV infection treatment and prevention. With the medication available now, individuals living with HIV can live essentially a normal lifespan. But I want to stress, especially for the young audience, and for the young people in the audience, that it was not always that way. In fact, it took almost 15 years since the start of the epidemics before reaching that stage. So during this period, we analyzed the function of this accessory protein by essentially generating viruses that were almost identical, except for the expression of very specific accessory protein. As I was mentioning before, we showed that they were dispensable for HIV replication. They displayed phenotype in primary target cells and some specific T cells, T cell line, but not all of them. And very interestingly, they appear to target a specific host protein, notably immunoreceptor, such as the CD4 receptor, as well as MHC class I molecules, which are so important in the recognition and elimination of infected cells by cytotoxic CD8 T cells. Hence, we and others showed that the VPU gene was augmenting HIV production by a mechanism that, by facilitating the release of HIV particle, as shown in the electron microscopy, by a mechanism that was unknown at the time, and that we know now that there is a restriction factor called tetrin, and I will talk about that in a moment, that VPU targets and degrade. We also showed that HIV downregulate the CD4 by VPU and NEF mechanism. And initially, we thought this process was thought to prevent superinfection, which is a process of rapid cell death. But as you will see, it turned out to be a very important immune evasion mechanism. Our work on VPR revealed that this accessory protein was virion-associated, pointing to a function, an early function of the protein in the virus life cycle, likely before de novo expression of viral protein. Remarkably, the expression of this protein induced a cellular growth arrest of cells at a stage of the cell cycle that is called the G2 phase of the cell cycle. And we showed that we provided evidence that that step was optimal for transcription for the HIV LTR. But importantly, we also showed that VPR was very important in facilitating and promoting viral replication in non-dividing macrophages by stimulating viral transcription. And we know now that the infection of macrophages is very important for pathogenesis and persistence. However, a key breakthrough, and I'm sorry for the complexity of that slide, but it's not the content is not so important. One of the key breakthroughs that advanced our understanding of HIV accessory function, as well as several steps of the viral life cycle, was the ability to perform whole genome screen using what we call small interfering RNA, or more recently, CRISPR-Cas9 technology. As you know, these technologies allow the knockdown or the depletion of very specific cellular genes or the knockout of very specific host factor, thus allowing the assessment of their contribution to HIV replication. These screens uncovered hundreds of host factors that HIV depended on to effectively replicate, what we called HIV dependency factor, HDF. This screen also uncovered cellular factors that were part of defense mechanism to inhibit or limit infection and replication, the so-called HIV restriction or resistance factors. So these studies demonstrated that the extent of HIV replication depended on a delicate balance between, on the one hand, the availability of numerous host dependency factors and pathways that had a positive effect on HIV replication, and on the other hand, cellular factors that had immediate antiviral activity and that are constitutively expressed by HIV target cells. These factors represent the first barrier to infection and dissemination and are shaping the initial stage of HIV infection, where the virus needs to amplify locally to establish infection and then disseminate in other tissues, such as the gut, that is very rich in CD4 positive T cells. These early defense mechanisms are, of course, complemented by innate and adaptive antiviral response that are generally inducible and are occurring later in infection. And here, I want to take a couple of minutes to highlight how these studies have led to basic discoveries that have changed our view of the viral life cycle, and at the same time have contributed to the recent development of a new class of antivirals that are targeting the HIV capsid. Indeed, the identification of key host nuclear transport factors, so factors that are very important for the transport of molecules between the cytoplasm and the nucleus, as well as combined with structural and imaging studies, have underlined the structural compartmentalization of the incoming virus particle, which really facilitate virus replication, but also allow the virus to hide from host innate and adaptive immune responses. This structural compartmentalization involves the mature capsid core that contains 2 copies of the viral RNA and which is made of a lattice of capsid proteins, hexamer and pentamer, at each of the ends, the yellow dots, that gives this very distinct shape to the capsid. When this core is released in the cytosol after viral entry, we know now that it binds a number of host dependency factors to facilitate its transport across the cytoplasm to the nucleopore, which are the gate of the nucleus. At the nucleopore, it binds to nucleopore proteins such as NUP153 or NUP358, and it is during this attachment that reverse transcription is occurring and the disassembly of the core to release the genetic content is happening. And part of the disassembled core is even directed through a partner called CPSF6, shows in green here there, to really direct the DNA genome to transcriptionally gene-dense region of the host for provirus integration. This structural compartmentalization is important for the virus not only to regulate its replication, but also to avoid the cytoplasmic sensors of foreign DNA that trigger antiviral responses. So why this is very important? It turned out that these advances have led to the development of the recently clinically approved anti-HIV drug lenacapavir, or GS6207, that was developed by Gilead. Lenacapavir is the first in-class capsid inhibitor with, as you will see, a multimodal antiviral activity. The development of this inhibitor is not only a chemistry accomplishment, but an example of rational design. The molecule binds, the molecule binds to a pocket of the mature core that you see here in the capsid lattice. And this site is involved in the interaction with this nucleopore protein such as NUP153 and CPSF6, which regulate capsid disassembly, nuclear import, and integration of viral DNA. So what GS6227 does is, as shown here, it really locks and stabilizes the core of the capsid, the capsid core, and basically prevents it to disassemble and release its genetic content in the nucleus for integration. In addition, at high dose, this compound has additional antiviral effect by affecting a late stage of the virus life cycle. It's generating aberrant particle, as shown in the electron microscopy here, that really inhibits its infectivity. The pharmacokinetic of the molecule makes it a very attractive, long-acting drug with proposed 6 monthly injections. And the molecule was FDA approved in 2022 for people living with multi-drug resistance, but there is clearly potential for its use in prevention, given its very high resistance barrier resulting from its multimodal mode of action. So now, regarding the defense mechanisms that are in place to limit infection and spread, we know now that lentiviruses have evolved to encode these accessory proteins to counteract the host restriction factors or innate immunity and adapt to cellular immune response. In these viral proteins are part of the many strategies that HIV use to persist despite antiviral response of the host. Indeed, CD4 T cells and macrophages are, contrary to what we thought initially, are not a very welcoming environment for HIV infection and replication. These cells express several restriction factors that are shown in orange and resistant factors that are shown in gray that are inducible by interferons and which can potentially suppress viral replication by acting at very specific stages of the virus life cycle. But by evolving to encode this accessory protein, HIV has the ability to neutralize and to overcome the antiviral activity of this restriction factor. Many of these restriction factors, and this is remarkable, are naturally expressed by T lymphocytes and macrophages and target areas of viral vulnerability that were used to generate antivirals. As you see here, this restriction factor is targeting the capsid. Several of these are targeting the reverse transcription, the nuclear import, as well as the late stages of the viral life cycle. Therefore, so much remains to be learned about these restriction factors, as they have the potential to inform and guide the development of HIV drugs with completely novel mechanism of action. Therefore, HIV has to overcome, as you see, several blocks to establish infection that can spread to different tissues, and this is in large part due to the ability of HIV to encode this accessory protein. Up to now, all these factors that are part of what we call intrinsic immunity have been identified at target step of the virus life cycle, either at the surface of the infected cells or in the cytoplasm. We know now of nuclear localized defense mechanisms that appear specifically to be there to prevent the expression of retrovirus, to silence them. One is the SMC5/6 complex that was recently identified, which interact with the unintegrated proviral DNA to silence viral gene expression from there before integration. And it is remarkable because Mark Wainberg was the first to show that you could have expression from an integrated DNA. Next, the other is the HUSH complex. The HUSH complex is an epigenetic remodeler that is recruited to the nascent viral transcript from integrated provirus and recruits a host machinery to silence HIV DNA that is integrated. And what is remarkable about the HASH complex is that it provides our intrinsic immunity with a way to recognize self and non-self integrated DNA by specifically silencing genes that have introns, that are regions that are not coding protein in our genome. But it turned out that HIV doesn't have intron, and that's why HASH has this specificity towards HIV and has the ability to silence it. These processes, which have been uncovered by the group of Paul Lehner at Cambridge, are the target of the lentiviral accessory VPR in the case of the SMC56 complex, and consistent with our initial observation that VPR was acting as a transcriptional level. And in the case of the HASH complex, it is the SIV and HIV2 VPX that are counteracting the HASH complex. So is there evidence of HASH repressing HIV, at least in vivo? So it is interesting. There has been remarkable studies from the lab of Matthias Lichtenfeld and Xu Yu, which provide evidence of that. The first paper deals with elite controllers, and more recently with people that have been on long-term ART for 20 years. What they found was that the intact proviral sequences from the elite controllers and some of the long-term ART controllers were integrated in specific, highly distinctive sites, preferentially centromeric satellite DNA or regions that are rich in CRAB ZNF gene family. This is very interesting because this is where HASH like to act. These sites are rich in epigenetic silencing marks and are heavily HASH repressed. So it is going to be interesting to determine whether these intact provirus are HASH repressed in elite and some long-term ART controllers. And it is interesting that the primate lentivirus family as a whole is able to counteract HASH. The only lentivirus that doesn't degrade the HASH is HIV, suggesting that HIV might exploit the HASH complex in the establishment of latency. The host complex HASH is not the only restriction factor that may impact the establishment of latent reservoir. Recently, in a very elegant study, Marc-André Langlois and Steve Barr at Western reported that even though the ApoBec 3F and 3G restriction factor act usually at the level of reverse transcription, they can impact HIV integration site selection by DNA editing of the end of the HIV LTR, which are so important for the integration process. And this condition, this editing, seems to promote latent infection, at least in vitro. Nicolas Chaumont and Pierre Gartner, in a landmark paper this year, have shown that HIV very rapidly, in fact, within the first 2 weeks, targets a diverse pool of CD4 T cells to establish productive and latent infection. Whether cellular restrictions such as SMC56, HASH, or ApoBec contribute to shaping the establishment of latent infection will be interesting to examine in the future. So how these accessory proteins counteract these restriction factors? This is an area where our lab has made important contribution, at least in the context of VPR and VPU. Remarkably, in the context of these viral proteins, were found to act as molecular adapters to connect and reprogram cellular function, mainly by recruiting protein degradation complex, which are called E3 ubiquitin ligases, or cellular adapter molecules, which are important in the trafficking of molecules in the cells for the purpose of neutralizing host restriction factors. While up to now, these were the most common mechanism to counteract restriction factor, recent evidence from my lab show that these viral accessory proteins do not always act directly on their targets. We recently uncovered a novel mechanism of counteraction by VPU, whereby VPU counteract a newly described restriction factor, which is called MARCH1, which inhibits HIV envelope glycoprotein packaging and viral infectivity by downregulating the expression of MARCH1 through the induction of what is called microRNA, which are very short microRNA that are able to bind messenger RNA by base complementarity and induce their degradation or block their ability to make proteins. Indeed, by recruiting a E3 ubiquitin ligase that targets a protein called beta-catenin for degradation, VPU increase beta-catenin levels and which drive the transcription of the MARCH1 targeting microRNAs. In addition to host restriction factors like serine 3,5 or tetrin, VPU and NEF target immunoreceptors such as MHC class 1 or such as TIM3, which was shown by Jimmy DeKaikos at Western to be downregulated by VPU, viral receptors such as CD4, and homing receptor like CCR7. So these proteins are completely remodeling the cell surface of infected cells. In the case of CD4, the antagonism by NEF and VPU was shown to prevent the unmasking of envelope glycoprotein epitopes, resulting in the accumulation of CD4 at the cell surface and the increased recognition of env glycoprotein by anti-HIV antibody, a process that triggers elimination of these infected cells by a process called antibody-dependent cellular cytotoxicity, or ADCC. So in the context of CAHR, we have harnessed this observation to develop new strategy to target and eliminate latent reservoirs. So in that context, Mario Ostrowski, using NEF blockers, has showed that by preventing the downregulation of MHC1, we could sensitize reactivated latent reservoir to cytotoxic T cell action and elimination. And we, in the context of my lab, in the context of shock and kill approach, we showed that CD4 accumulation and variant tethering by the restriction factor tetrin was sensitizing infected cells and reactivated latently infected cells to this process of ADCC. Similarly, the laboratory of Andres Finzi and Joe Sodrowski have shown that a novel class of molecule developed by Amos Smith at Harvard called CD4 mimetics mediate potent sensitization of infected cells, again, to antibody-dependent cell cytotoxicity, ADCC, thus underlining the potential of harnessing what we have learned from the function of these accessory proteins for the development of strategies to clear viral reservoirs. So in conclusion, investigating HIV host interaction at the cellular and molecular levels continues to be instrumental in achieving practical clinical outcome. HIV has evolved to encode accessory proteins, to reprogram basic cellular pathways to counteract host intrinsic and innate immunity and adapt immune responses. These viral proteins contribute to the immune evasion mechanism that allow HIV to persist in a host, particularly during the acute infection. And the interplay between this host restriction factor and accessory protein is providing insight into the establishment of latent viral reservoir and is informing and guiding the development of cure or remission strategies. So the message that I would like to make to the young people in the audience, the graduate students, the postdoc, and the young investigator, is that all this basic work that is driven by curiosity and a quest of understanding how things work open new perspectives that can lead to the implementation of something that is clinically actionable, as was demonstrated with the development of lenacapavir. That is why we need to pursue basic scientific advances and the implementation of breakthrough if we want to see the end of the HIV/AIDS pandemic, either with the preventive tools we already have or by, hopefully, the development of an effective or a cure. And retrospectively, given the influence that Mark Wainberg had on the generation of HIV scientists in Canada, this is going to happen if we continue his legacy to support young trainees, pre-doc, postdocs, and young investigators. It is going to happen unless these young people in the audience will do it. So, as Tony Fauci nicely put it at CROI 2023, please take the ball and run with it. So I would like, in the final note, to really thank all the trainees and staff that have driven the scientific program of my lab in the last 30 years. Of course, the many collaborators that interacted with our lab, particularly the member of CAHR, the member of the CHAMP CRHR team, and of course, all the founders that allowed our scientific program to progress. So I want to thank you for your attention and enjoy CAHR 2023 conference. Thank you.
Tiens, Éric.
Merci.
Picture? Yes. Il faut qu'on fasse une picture. Ah oui, alors, je fais figure souvenir.
Comment ça?
Oh, yes, with the award.
Ah bah oui, oui, oui. Il avait caché.
Thank you. Thank you.
Thank you. Thank you.
Merci, Éric.
Thank you very much.
Oh, oui, oui. Donc merci Éric pour ces 30 années de résumé finalement en très peu de mots. Maintenant, nous allons en fait conclure cette première séance et nous allons vous inviter donc à nous rejoindre, Cecilia et moi, au niveau de la réaction d'ouverture. Encore une fois, remercions en fait le CAHR, ses organisateurs qui nous ont permis aujourd'hui. On a été pas mal sollicités, c'est vrai. On court toujours contre le temps, mais merci quand même encore à vous. Et puis merci tous d'être ici. Encore une fois, plus de 670 personnes. 350 résumés. Ça démontre un petit peu aussi le dynamique. Souvent, on dit le sida, c'est derrière nous maintenant. Hein. On pense qu'on a tout résolu, mais il reste encore beaucoup de chemin. Merci à tous.
So thank you, Dr. Cohen, for the very engaging and inspiring speech, very much like Mark's speeches used to be. As Jerome says, we'd like to thank all the participants and everyone for coming out. We have over 600 registrations and over 300 abstracts submitted, which shows the dynamic nature of all the research which is going on today. Before we head over to the poster and exhibit area, we just have a few housekeeping things to remind you of. You can download the conference app, and the information is here, and it's also on the website and in your programs. This is the password to connect to the Wi-Fi network. And we'd very much appreciate if you could please complete the session survey, and this is available as well on the conference app. So now this concludes the opening reception, or at least this part. We'll now move over to the exhibit and poster area, and we wish you all a wonderful conference.